Combinatorial chemistry using microdroplet libraries is a cutting-edge strategy that combines the principles of chemistry and microfluidics to rapidly generate and screen vast arrays of chemical compounds. In traditional combinatorial chemistry, chemists synthesize large libraries of compounds by systematically varying reactants and reaction conditions, then test these mixtures for desired properties such as biological activity or catalytic efficiency. By incorporating microdroplet technology, researchers can miniaturize and parallelize these reactions, creating thousands or even millions of tiny reaction vessels, each a microdroplet, that serve as independent laboratories. This approach dramatically increases throughput, reduces reagent consumption, and allows for more precise control over reaction conditions. Imagine a large library where each book represents a unique chemical compound. Traditional methods would require reading each book one by one, which is time-consuming and resource-intensive. Now, envision that each microdroplet is like a digital book that can be instantly accessed, analyzed, and compared with others simultaneously. This is the essence of microdroplet libraries. They enable rapid screening of chemical diversity by encapsulating reaction mixtures in droplets that are uniform in size and composition, ensuring that each book in the library is produced under nearly identical conditions. The use of microfluidic channels and droplet generators ensures that these droplets are highly monodisperse, meaning they have a consistent volume, which is critical for reproducibility and quantitative analysis. The science behind this approach involves a careful orchestration of fluid dynamics and reaction kinetics. In a microfluidic device, reactants are introduced into microchannels where they meet at precisely designed junctions, such as TJ junctions or flow-focusing geometries. Here, the aqueous solutions containing different reactants are segmented into droplets by an immiscible oil phase that acts as a carrier. Each droplet acts as an isolated microreactor where controlled chemical reactions occur. For example, if you are trying to synthesize a library of novel catalysts, you might combine various metal ions, ligands, and reducing agents within each droplet. As the droplets flow through the channel, reaction conditions such as temperature and concentration can be finely tuned by adjusting flow rates or by incorporating temperature-controlled zones. This level of control allows chemists to explore a vast chemical space systematically and efficiently. One might ask, why is it advantageous to perform combinatorial chemistry in microdroplets compared to bulk reactions? The answer is multifaceted. First, the miniaturization of reactions in microdroplets significantly reduces the volume of reagents needed, which is both cost-effective and environmentally friendly. Second, because each droplet is isolated, there is minimal crosstalk between reactions, ensuring that the outcome of one reaction does not interfere with another. This isolation is crucial when screening for specific activities, such as drug candidates, where even a small amount of cross-contamination could lead to misleading results. Third, the high-throughput nature of microdroplet libraries means that thousands of reactions can be performed in parallel, greatly accelerating the discovery process. To draw an analogy, consider traditional combinatorial chemistry as akin to painting a vast mural one brushstroke at a time, with each brushstroke representing a different compound synthesized in bulk. In contrast, microdroplet libraries are like having a digital palette where every possible color and blend is created simultaneously, and you can quickly evaluate which combinations produce the most vibrant or desirable effect. This simultaneous creation and screening of compounds are what make microdroplet-based combinatorial chemistry so powerful. A practical example of this technology can be seen in drug discovery. Researchers can encapsulate a variety of potential therapeutic compounds into microdroplets along with a target biomolecule, such as a protein involved in a disease pathway. As the droplets incubate, any interaction between the synthesized compounds and the target can be detected by changes in fluorescence or other optical signals. This rapid screening process helps identify lead compounds that show promising activity, which can then be further optimized and developed into drugs. Similarly, in materials science, microdroplet libraries enable the synthesis of novel polymers or nanomaterials by varying monomer compositions and reaction conditions in parallel. By screening these materials for properties like conductivity, strength, or optical activity, scientists can rapidly discover new materials with tailored functionality. Another exciting application is in catalyst discovery. Traditional methods for finding efficient catalysts often involve laborious testing of numerous reaction conditions. By using microdroplet libraries, chemists can vary the type and concentration of catalysts, ligands, and substrates in thousands of individual droplets, each acting as a mini reaction vessel. High-throughput screening then identifies which droplets yield the best catalytic performance. This approach can uncover novel catalysts that might have been overlooked in bulk experiments due to subtle differences in reaction conditions. One may wonder, how do researchers manage and analyze the enormous amount of data generated from these high-throughput experiments? The answer involves the integration of advanced optical detection systems and sophisticated data analysis algorithms. Fluorescence microscopy, mass spectrometry, and other analytical techniques are used to rapidly characterize the outcome of each droplet reaction. Coupled with automated image processing and machine learning algorithms, these tools can sort, quantify, and analyze data from thousands or even millions of droplets in a relatively short time. Imagine having a supercomputer that can instantly read, compare, and catalog every book in our digital library analogy. This is essentially how data from microdroplet libraries are processed to identify the most promising compounds or materials. Furthermore, microdroplet libraries are not static. They can be dynamically manipulated to explore reaction kinetics and optimize reaction conditions in real time. For instance, by using techniques such as pico-injection, additional reagents can be added to pre-formed droplets at specific time intervals, allowing for multi-step reactions to occur within a single droplet. This sequential addition of reagents is analogous to following a multi-stage recipe, where each step is carefully timed and executed to achieve the perfect final dish. Such dynamic control over reaction conditions is crucial for exploring complex chemical spaces where the outcome depends on precise timing and concentration profiles. A common question that arises is, what are the limitations of using microdroplet libraries in combinatorial chemistry? While the advantages are numerous, challenges remain. One major challenge is ensuring the stability and uniformity of droplets over extended periods, especially when reactions require longer incubation times. Droplet coalescence or evaporation can affect reaction outcomes, so researchers must optimize surfactant formulations and device designs to maintain droplet integrity. Additionally, integrating multiple steps of a reaction within the confined space of a droplet requires precise control and synchronization, which can be technologically demanding. However, ongoing advancements in microfluidic chip design and automation are continuously addressing these challenges, making the technology increasingly robust and scalable. Looking forward, the future of combinatorial chemistry using microdroplet libraries is exceptionally promising. With improvements in microfabrication, detection technologies, and data analysis, it is likely that even more complex chemical libraries can be generated and screened at unprecedented speeds. This will accelerate the discovery of new drugs, materials, and catalysts, potentially revolutionizing multiple fields. Imagine a future where chemists can design and test millions of compounds in a matter of hours, dramatically shortening the time from discovery to application. A future where the potential of microdroplet libraries is fully realized. In summary, combinatorial chemistry using microdroplet libraries offers a powerful, high-throughput, and highly efficient method for exploring vast chemical spaces. By partitioning reactions into thousands or millions of uniform microdroplets, researchers can rapidly synthesize and screen diverse compounds with minimal reagent consumption and high precision. Whether applied to drug discovery, materials science, or catalyst development, this technology is transforming how complex chemical reactions are conducted and analyzed. The integration of advanced microfluidic techniques, real-time monitoring, and sophisticated data analysis tools is paving the way for breakthroughs that were previously unimaginable.